you're going to hear about tons of different tissues and systems throughout your body. Um, but I'm the one that gets to talk to you about blood groups, what's actually in our blood. So, one of the things that people often know about themselves when they can rattle off statistics about themselves, like name and address and all of that, is their blood type. So, for example, I am O positive. So, if I ever wind up in your ER, keep that in mind. But in the meantime, what does that actually mean? What does that blood type actually tell you? And why is it important clinically? And what kind of responses do we make to it? So that's what we're going to go through in this video. And then at the end, I'm going to do kind of an overview of two types of reactions, precipitation and agglutination. And the reason is because those types of reactions are heavily involved with blood groups in general. All right, so in general, there are three main groups for blood types: type A, type B, and type O. So what does this actually mean? So the blood groups, the blood group antigens, which is what these are actually carbohydrates. So these are carbohydrates that exist on the surface of our red blood cells, okay? There are three different types of them. O, and then an A and a B. The O carbohydrate is basically the simplest carbohydrate structure. Okay, so you can see it has a pink ring, a yellow ring, a green ring, and a yellow ring. All right. A and B share the same O structure, as you can see here, but they have added side chains that make them different. Okay, so O is the base structure, and A and B are accessorized. All right. So when a person has O blood type, that means they have kind of the baseline carbohydrate structure on their red blood cell surface. So the other way of putting it is they have no antigens present because they have no like different antigens present. They just have kind of what's expected, the factory design, all right? Patients who have A blood type have these carbohydrates on the surface of their red blood cells. Patients who are B have these on their blood cells. And patients who are AB have both, okay? So that means the antigens present on the red blood cell are listed here. You can either have A antigen, B antigen, A and B, or none. And none, again, is just O, which is kind of the factory design, all right? Now, let's talk about what is made to these. First off, I already told you that these were carbohydrates, all right? These are carbohydrate antigens. So that means that if we're going to make an immune response to any of them, it has to be IgM, right? Because in order to get to any other type, isotype of antibody, we would need T cell help. T cells only see peptides. Since these are not peptides, we're only going to make IgM, all right? And you can see that's what's shown here. You've got these little pentamers indicating IgM. When we're exposed to different blood types than our own, we're going to make an anti blood type antibody, and that antibody is going to be IgM to that blood type, okay? So for example, I'm type O, I receive, say, type A blood, I'm going to make anti-A antibodies, all right, to that blood type. If I was type B, I would make anti-A antibodies. If I was type A, I would make anti-B antibodies if I was exposed to type B blood. So you're going to make antibodies to the blood types that you are not, all right? And that's why group AB is kind of part of the universal acceptor group, right? Because they have antigens for A and B. So they can receive type A blood or type B blood and not make any immune response. Type O, therefore, becomes the universal donor. I can give my blood to pretty much anyone, and they're going to be okay with it. There's an exception to that, and I'll explain it in a minute, all right? So they're going to make these antibodies. Now, how do we make these antibodies, and why do we make them? This is actually a great example of cross-reactivity, all right? So let's think about what these antigens are. These are carbohydrate antigens. What we've figured out is that these carbohydrate antigens are actually also expressed on microbiota found in our gastrointestinal tract. Okay, so they're shared. So that's part of the reason your body thinks that this is some foreign antigen. And it is, it's not self to you, but it's not harmful, right? It's just another human antigen. But because we also see them on the microbiota that we find in our gastrointestinal tract, we might make a response to them. These antigens are referred to as isoagglutinins. And the ones that are found on the microbiota are so similar to our own type A and type B antigens that we might respond to them if we saw them. Now, if you are type A blood, the only reason you don't make an immune response to these carbohydrates when they're on microbiota is that you're tolerant to them. So since it seems like a self-antigen, your body's like, not that one. Don't respond to that one. Since it doesn't see the B antigens, because you're type A, when it is exposed to type B blood, it'll be like, that one, kill that one. Whew, sorry about that, guys. That was the fire alarm. So we just all went downstairs, stood outside, and then I climbed 5 flights of stairs back up. So a little out of breath. But anyway, back to blood groups. So microbiota are basically what we are responding to. We will be tolerant to the antigens that we express as self. So if you're type A, you'll be tolerant to the type A microbiota, but intolerant to the type B microbiota. If you're type O, you'll be intolerant to both. So that microbiota will mount an immune response and then probably not be part of your normal commensal microbiota. So think of it this way, and I really like this table because I think it displays it nicely. So if you're group O, you are intolerant to A and B. If you're A and B, you're tolerant to everything. If you're B, you're intolerant to A, and if you're A, you're intolerant to B. Now, what actually happens? When you make these, remember that IgM is really good at stimulating complement. And complement is super pro-inflammatory. So basically, you're going to begin the process of complement, which will lead to destruction of red blood cells that do not match your own. So when you have a bad type and cross or a bad cross match, when you give a patient blood that they are intolerant to, you can get hemolytic anemia of that transferred blood, which presumably you are only giving to them because they need blood. Okay, but what about positive and negative? So earlier when I shared my blood type with you, I told you I was O positive. So we have people that are O positive and we have people that are O negative. So what does that mean? Okay, that positive or negative is indicative of a protein antigen that's known as the rhesus antigen or RH factor. Okay, it's inherited from mom and dad. So let's make ourselves a nice little Punnett square. Okay. And let's say on top, we're going to have mom, and on bottom, we're going to have dad, okay? Let's say mom is O positive, okay? And I know that my father was O positive and my mother was O positive. So it is likely that I am positive for RH factor. And let's say that dad is A negative. So that means he has to be negative, okay? So then all of our kids would be heterozygous for RH factor. So what does that mean? That means that all of our kids are going to have a positive blood type. So if I'm O and my husband is A, our kids would be A positive. Does that make sense? All right. I know you can't answer me. I'm really used to asking that question in front of people, but I appreciate if on the other end of this video you're nodding. Anyway, let's say that then one of these kids goes out and has kids of their own, right? So we've got a positive, negative, and let's say we've got somebody else who's a positive, negative. So they have a chance at having kids that are positive, a chance at kids that are heterozygous for RH factor, and a chance of kids that are negative, okay? Only this one will result in a negative blood type, right? So if you had an A positive and a B positive person with this kind of heterozygous combination who wound up here, you would have a B negative blood type. Okay, so it's a standard Punnett square. Now, it's a protein. That's important to note. If it's a protein, that means we can class switch. So the isotype of antibody made to this one is IgG. IgG is also really good at activating complement. But the other thing that IgG does is cross the placenta. And so that's actually a big problem in pregnancy, and we'll talk about that in a minute. Now, I do want to say one other thing about babies and blood types. Remember that these O, A, and B antigens we only make antibodies to them when we're exposed to food, all right? When babies are first born, their microbiota is very limited, and they haven't really made any antibodies yet, right? The only antibodies they have for those first couple weeks of life are the ones they got from mom in utero. So if you had like a 2-week-old baby who desperately needed a blood transfusion and you don't know the baby's blood type, what do? You have to know it, right? Wrong. Because they have not had the opportunity to make antibodies to those isoagglutinins, technically a very, very young newborn could receive any blood type and it would be okay because they don't have any responses made. It's not a risk I'd want to take, but it's kind of cool when you think and conceptualize out how we make responses to blood types. All right, so right after I got finished telling you that babies are safe from bad crosses of blood, let's talk about how blood reactions are actually really bad for babies. This is known as hemolytic disease of the newborn. This is a really actually excellent example of a type 2 hypersensitivity disease. The severest form of hemolytic disease of the newborn is known as erythroblastosis fetalis. And that actually results in death of the fetus. In this case, what happens is a mom who is Rh negative, so let's say an O negative mom, has a baby with somebody who, let's just keep it simple, is O positive, all right? That means the fetus is going to be O positive, right? So during the first pregnancy, this is probably not a problem. But during pregnancy, as the baby's red blood cells turn over, which happens, mom gets exposed to these Rh antigens. So she starts making antibodies to Rh antigens. This typically happens at birth, but it can happen during pregnancy, all right? So now the baby is born and the baby is fine. First pregnancy, no problem. But mom has made these anti-Rh antibodies. Now this couple decides to have baby number 2, who is going to be O positive. These anti-Rh antibodies are IgG. That means they can cross the placenta. And what happens is they cross the placenta and they attack the baby's red blood cells. And that's what leads to fetal loss. Basically, it's a hemolytic disease of the fetus. This isn't always fatal. Let's think just quickly about the mechanism. Remember, this hemolytic disease is actually due to complement. And complement is an innate immune response, which means we're born with it. This doesn't require growth like development of antibodies. Mom's antibodies are stimulating complement that's causing death of the fetus. So when is it not fatal? Sometimes babies are just born jaundiced. And the reason for this is that as the red blood cells break down, the bilirubin is not adequately broken down. When the baby is still in the womb, bilirubin actually crosses the placenta and is basically removed by mom's liver. But when the baby is born, the baby can't really keep up with all this bilirubin. So you get this accumulation of bilirubin, and that can actually be diffused to the brain and cause an irreversible damage known as kernicterus, and in some severe cases can kill the baby. However, if the baby is just jaundiced, actually what we do is we put them in these little baby tanning beds. Isn't that cute? And that'll actually break down, the UV light actually breaks down the bilirubin. The bilirubin is cleared, the antibodies go away in time, and baby is fine. Even better than little baby tanning beds is actually RhoGAM. And RhoGAM is actually what we can do to prophylactically treat RH mismatch. Hemolytic disease of the newborn can be completely prevented by treatment of the RH negative mother with RhoGAM. This is just a preparation of human anti-RHD IgG antibodies. So basically you're taking IgG antibodies that are specific for RH, and you're injecting them into mom at 28 weeks of gestation, and then again 24 hours after birth. And if you do this, it basically eliminates the fetal RH positive red cells before they can generate an RH specific memory B cell response in the mother. We don't entirely know the mechanism by which it works, but we do know that that's the end result and that it means that it's no problem for the fetus. Even better than this would actually be diagnosing it first. So let's talk about how we diagnose when there are antibodies to blood cells. Okay, these two tests can be used to detect antibodies to blood cells in a variety of situations. So we could use it in hemolytic disease of the newborn or diagnosis of that potential, but we can also use it for like acute hemolytic anemia, which can happen for a bunch of different reasons that we'll talk about. Recent penicillin use is one of them. Some people also begin to make antibodies for other reasons. So these two tests are basically used to identify antibodies in the bloodstream and antibodies already bound to red blood cells. So this is known as the Coombs test. That's kind of what it was historically known as. Now it's known as either the direct antibody test, or DAT, or there's another one that's the indirect antibody test, or IAT, okay? These tests, like I said, will detect the presence of antibodies that are either already bound to red cells or antibodies that could potentially bind to red cells, all right? So we're going to start with the direct. The direct means the antibodies already bound to the red blood cells. So what we do is we take a blood sample from a patient with who we suspect of having a hemolytic anemic event. So we take their red blood cells. Note that in this patient, for the result to be positive, the antibodies are already bound to the red blood cell inside the patient. So we're going to take these and we're going to mix them with a reagent. And this reagent is actually antibodies that are specific for this antibody, all right? So we're taking the actual red blood cells themselves, we're mixing them with the reagent and seeing if the red blood cells agglutinate at the bottom of the well. If they do, when you have the isolated red blood cells and the reagent, then that means you have antibodies that are already bound to the red blood cells. Now, let's say we want to determine if there's even the possibility that there are antibodies that could bind to a red blood cell antigen. In this case, we're only going to take the patient's serum. So we still take a blood draw from the patient, but then we spin it down or we let it settle and we remove just the serum because antibodies are found in the serum. We then take the donor's blood sample and we add it to the tube with the serum, okay? The recipient's serum is going to, this is when you're trying to do a mixed leukocyte reaction. So basically you're taking your recipient and you're taking their serum and you're going to add it to red blood cells to see if they're going to react. People often use red blood cells. You could also use latex beads that are coated with your antigen. So this is a different person's blood cells or latex beads coated with antigen. You're going to mix those in a tube, okay? So the different red blood cells or the different antigen that is coated on the latex beads with the serum from your recipient. You're then going to add that same reagent from before and see, did these antibodies bind these cells? Because if these antibodies bound these cells, when the reagent is added, will create this lattice and it will agglutinate. And that's the IAT. So IAT means there are antibodies specific for that antigen in the serum, but they have not bound that antigen yet. DAT means the antibodies have already bound their antigen on the red blood cell. All right, I just want to take a minute to differentiate agglutination versus precipitation. Responses made to red blood cells are a really great way to differentiate this because they occur as a result of immune complexes, which is basically antibodies binding antigen. And the form taken by the antigen-antibody complex depends on the nature of the antigen and the ratio of the antigen-antibody concentrations, okay? So agglutination is particularly associated with red blood cells and latex beads. If you're reading a step 1 STEM that asks you about red blood cells and latex beads, and then asks you, did this agglutinate or precipitate? The answer is always agglutination. If you see red blood cells or latex beads, it's agglutination. If you do not see red blood cells or latex beads, that means it's everything else and it's precipitate. Everything else precipitates. Only red blood cells and latex beads agglutinate, okay? Precipitation of an immune complex can be observed when there's increasing amounts of antigen in a series of tests. So as we get to antigen excess, we see precipitation. When we have antibody excess, we're more likely to see agglutination, okay? So antibody excess, the concentration of antibody is in excess relative to the concentration of antigen. So generally, the antibody is bound in kind of a univalent fashion, and you can see kind of these lattices forming because they're kind of forming together, okay? We'll also see that at equivalence when the amount of antigen and antibody is equivalent, you see this bivalent binding, and that forms a nice lattice as well. And that can lead to kind of this lattice form that can also agglutinate. But antigen excess, that's going to be precipitate. You have so much antigen that all of the antibodies are bivalent and they're not associated with each other, okay? All right, so what's the clinical significance of this? You'll learn about this more when you think about type 3 and type 2 hypersensitivities. But remember, an immune complex is just an antigen that has already bound its antibody, okay? Disease will typically occur where immune complexes deposit, and that's because the immune complex itself can activate complement, and complement is super potent. If we're talking about type 3 hypersensitivities, which is pretty much where I worry about immune complex deposition, you're going to want to track serum C3 and C4 components, because what happens is that complement gets activated so much over and over and over again that eventually we actually see a depletion in C3 and C4 because we're using up the components faster than our livers can make them. You can also just check to make sure that complement is functioning correctly using the CH50 test, which basically just tells you whether or not it's functional. It's the functional integrity of the entire classical complement pathway. It also might be reduced due to consumption of classical pathway components. So like if you're really using up your C3 and C4 like you would in a type 3 hypersensitivity reaction, your CH50 might be decreased as well.